In this third video clip from chapter 13, we're going to now begin to look at something called X inactivation. So although female mammals inherit two X chromosomes, one of the X chromosomes can become almost inactive during embryonic development. This condensed chromosome is known as a Barr body, and therefore most of the genes on the Barr body would not be expressed. Now, we can look at this to explain why tortoiseshell and calico cats are always female. If we look at an early embryo, in this case where the cat has an allele for orange fur and an allele for black fur on the X chromosome, after a few cell divisions, the X inactivation occurs, and we end up with two different populations of cells, one expressing an allele for black fur and one expressing an allele for orange fur, because the other one, the Barr body, is inactive. And so you end up with cat coloration called tortoiseshell with this sort of mottled black and orange fur. Now, calico cats actually have a gene for a third color thrown in there. And so it's for this reason that calico cats are always female. Now, changes in chromosome number or structure can also cause some genetic disorders. Typically, plants can tolerate more large-scale changes to chromosome structure and number than in animals. In fact, many hybrid varieties of crops have extra chromosomes. They're polyploidy, and this is to produce desirable characteristics. And we'll talk more about hybrids in 1407 next semester. Now, large-scale changes in chromosomes often lead to spontaneous abortions of the fetus in animals, while smaller-scale changes to chromosome number can simply affect the phenotype. And we call these small abnormal chromosome number changes, this is referred to as aneuploidy, where we have an abnormal number of chromosomes. Now, if a cell has one extra chromosome, diploid plus one, it's said to be trisonomic because there would be three. If the cell has a chromosome missing, therefore it's diploid number minus one, it's said to be monosomic because it would only have one. And most often these mishaps are caused by something called non-disjunction that occurs during meiosis, where parts of homologous chromosomes or the sister chromatids don't separate the way that they should. And this is a good time to review meiosis anyways. Remember that meiosis includes two divisions of the cell in cytoplasm, meiosis 1 and meiosis 2, to produce our gametes. A non-disjunction occurs when either the tetrads do not separate as they should during anaphase 1, and notice here where the tetrads are joined together at the chiasma as a result of crossing over, this connection is maybe incorrect. It has strengthened over time. It doesn't allow the tetrad to be pulled apart, so the tetrad moves in this direction, which leaves us missing some chromosomes in this cell and having some extra chromosomes in this one. So we end up at the end of meiosis 2 with half the gametes that are monosomic and half the gametes that are trisonomic. While a non-disjunction that occurs in meiosis 2 occurs as the sister chromatids don't separate as they should because of the connection or the centromere. And if the non-disjunction occurs in meiosis 2, half of the gametes are completely normal, right? If we follow this lineage of cells, these two gametes are completely normal haploid. But it's these that are missing one, or monosomic, or contain an extra one that's trisonomic. Now, we can look at some human disorders due to things like this, and probably the most common of which is Down syndrome, also known as trisomy 21. If we look at this karyotype, we can see that this individual is trisonomic, 1, 2, 3 chromosomes for the 21st pair. Individuals with Down syndrome have very characteristic facial appearances. They're very short in stature. Their cardiovascular system and their heart may have some developmental effects. They may not be fully developed mentally or sexually. Now, the odds of this non-disjunction increase in mothers over the age of 30, and this has to do with the way that female gametes are produced during embryonic stages, and then they are sort of in a state of suspended animation from birth until puberty, when once every 28 days during the cycle, a new ovo can be produced that will actually continue meiosis. Until then, these cells are sort of in this state of suspended animation. And over this length of time, these connections can be stronger. So the length of time after puberty has an effect on the probability of non-disjunction occurring. And then we can also look at aneuploidy of these sex chromosomes. If an individual has an extra X chromosome, so this would be an individual that's trisomic for the 23rd pair with an extra X, they exhibit something called Klinefelter syndrome. These individuals are phenotypically male, but they are sterile. While an individual that is monosomic for the 23rd pair, missing one of the chromosomes, missing the X, it has Turner syndrome, and again, they are typically female and sterile. Now, a lot of the genetic disorders that we've talked about in the old days were noted due to some genetic testing and counseling. Several years ago, to obtain cells for genetic testing, the cells were obtained from the fetus by something called amniocentesis, where a very, very large hypodermic syringe was carefully inserted into the uterus, and some of the amniotic fluid that surrounds the developing fetus and contains some of the fetal cells was drawn up, and these fetal cells could then be analyzed. This was obviously a very intrusive procedure, and you oftentimes couldn't tell the fetus to be still and not move. Now, a much less invasive method is something called chorionic villus sampling, and this is where we insert a tube using an ultrasound device that samples some of the cells from the chorion. These cells are fetal cells, and they could be tested genetically. There's also some non-invasive parental testing nowadays that can be done using biomarkers found in the blood plasma or fetal DNA in the maternal plasma. And it brings some interesting questions up, about the different types of genetic diseases and so forth. Many individuals who have history of these diseases might consider being tested to determine the likelihood that they could have them or pass them on to their children.